Good afternoon, ladies and gentlemen. I would like to welcome you to my presentation about the production of microbial biosurfactants for cosmetics and detergent industry. Surfactants in chemistry are molecules with a surface active property. They can emulsify water and oil, which is used in food industry, cosmetics, pharmaceutical, or crude oil recovery. Surfactants has also the ability to detach oil from solid phases, and this is the reason why they are also used as detergent in cleaning agents like washing powder, but also in products for personal care. We have a high demand on surfactants. This is the reason why in Europe 2.5 million tons are produced. Mostly the surfactants are used for the production of cleaning agents, what is highlighted in green. Or they are used also in a higher amount in cosmetics or textile industry. This is highlighted in orange and blue. If we have a look which raw material is mostly used for the production of surfactants, then we could notice that 50% are based on fossil raw material like crude oil. And just 7% of all produced surfactants are fully bio-based and produced with renewable resources like sugars or plant oils. In this slide, I want to show you two bio-based surfactants that are produced by microorganisms and are commercially available. The first molecule is sulforolipid, what can be produced by yeast strains. The second molecule is rhamnolipid, which is produced by bacteria. Both molecules belong to the glycolipids because they have a hydrophilic sugar part and a hydrophobic lipid part. In my research group, our aim is to optimize processes for the production of two other kinds of biosurfactants, and these are not yet available in a high amount on the market. These biosurfactants are called Monosule eretritolipids or Cellobiose lipids, and both can be produced by Fungi strain in a bioreactor. For the production, we just need to feed sugars or sometimes also plant oils. In the following, I would like to give you an insight into what the Monosule eretritolipids are suitable for. We have found and analyzed 265 application patents. Here we found that more than half of them are patents for cosmetic applications. We have further read patents in the field of detergent, pharmaceutical applications and plant protection. Interestingly, only 35% of the patents are active and only 6% of them are valid in Europe. This now offers many companies the possibility of freedom to operate. Now I will come to some examples of defined properties. Cell activation is described as a concrete application example in the literature, as the molecule has a ceramide-like structure. For the Monosule eretritolipids, also self-assembling behavior and protein binding capacity is shown. In addition, the Monosule eretritolipids can also have an anti-inflammatory effect or moisturizing and skin protection properties. Let me introduce first the molecule structure of Monosule eretritolipids. This molecule has a backbone of one sugar molecule, mannose, the sugar alcohol, erythritol, and they can have two lipid chains. The molecule can take on different variations. For example, the side chain groups on the C4 and C6 atom can take an acetyl group or hydroxy group. Due to this, we can have 4 different variants that leads to different physicochemical properties, like polarity or water solubility. Another variability are at the fatty acid chains. They could have a long chain, a middle, or also a short chain, what has also a high influence on their final application properties. In my research group, we have tested different fungi strains, what are highlighted in green, and showed that they are producing different molecules, which gives the industry the opportunity having a wider range of surfactants for their application. For the production of these biosurfactants, Monosule eretritolipids, we optimized for several of these microorganisms a fermentation process and also a purification process with the possibility to produce a high quality of Monosule eretritolipids. For the purification, we need first to separate the cells and then also separate the water and the remaining oils and fatty acids from the feeding solution. On the right-hand side, you see a picture with our pure mel, which looks honey-like and is a liquid with a high viscosity. I now come to the Cellobiose lipids. It is known that they show gelling characteristics and are effective against biofilms. Cellobiose lipids have an antifungal effect. In the field of plant protection, it is shown that Cellobiose lipids have an effect against cucumber powdery mildew. For the Cellobiose lipids, we have found and analyzed 112 application patterns. Most of them are in the field of cosmetics or detergents. We have mentioned that just 42% of the patents are active and only 5% of them are valid in Europe, and thus also offers the possibility of freedom to operate. In this slide, I want to introduce you the next biosurfactants we are working on. They are named Cellobiose lipids, and dependent on the microorganisms, also different molecule variants can be produced. The most differences are in the side chains at the sugar group, and this changing has also an important influence on their final properties. However, it is also possible to obtain further variability in the molecular diversity by inducing it chemically or enzymatically. On the left side, you can see how we were able to cut off the side groups by treating with alkaline solution. On the right side, we were using enzymes and was able to carry out an esterification. In this slide, you can see a laboratory bioreactor where we optimize the production of Cellobiose lipids. Here we just fed sugars to the fungi. After purification of the fermenter broth, we was able to produce white Cellobiose crystals. This you can see in the picture on the right side. In the last slides, I showed you that we were able to produce different molecules, and interestingly is that all molecules have a very good ability to reduce the surface tension by the use of a low amount of milligram per liters. But we have also observed that the molecular structure can also fundamentally change the properties. For example, we have now the possibility to produce biosurfactants that have a foaming effect, and also those that do not foam. There are applications for both types of the surfactants. For example, foaming is undesirable in a dishwasher, but in the field of hair shampoo, one would like to have a high foaming effect. In this slide, I would like to present one of our projects, the Biosurfactants Alliance. We work together with different companies and universities in Germany to optimize the production of biosurfactants with different residues and to analyze the application characteristics in the field of detergent and cleaning agent, cosmetics, bioremediation, crop protection and food. In addition, we make the holistic assessment for the economic and ecological feasibility of the production process. We work together with the companies Pfeiffer & Langen, Hermetia, Festo, Biotensidon, BASF, Analyticon, In Silico, Dali and Henkel, and the universities in Stuttgart, Karlsruhe, Hohenheim and Munich. Last but not least, I would like to summarize once again that both Monosule eretritolipids and Cellobiose lipids have a very great potential for many applications in industry due to their structural diversity. In our research, we were able to identify many structures and optimize the process to such an extent that we can produce highly pure surfactants. In the coming years, we will continue to work on process optimization and scaling up. If you're interested in testing these surfactants, we would be happy to operate with you. I'm at the end of my presentation and would like to start by thanking Jasmin Schubert and Katharina Grefe for inviting me to give this talk. I would also like to thank my working group and the great scientists and also the donors for the financial support. And finally, I would like to thank you for your attention.
So thank you very much for this very interesting presentation on biosurfactants, and hopefully Susanne is already in the line. So welcome to our studio.
Yes, hello Achim, hello to the audience. So thank you for inviting me.
Okay, like in the other presentations, the audience has a chance to set questions in the Mentimeter. I invite everyone to do so, and I will start with a more market-oriented or product-oriented question. So, where are the first uptakes of biosurfactants, for example, for sophoro and rhamnolipids? So, are these already on the market somehow?
So they are on the market, so they are available. So there are companies that they are producing sophorolipids and rhamnolipids. And to be honest, I just know one product. So I'm not sure if they make advertisement out of it, but there's one product, what is, I hope, what is from the company Ecoware, what has included the sophorolipid. But I guess there are already different others also available.
Okay, this is a cleaning agent as far as I know, and I think there are also some in the cosmetics area linked to Evonik. But then directly the next question, you are working on other kinds of biosurfactants. So where do you see the market opportunities for these new kinds of biosurfactants compared to the, I think I say more traditional more traditional surfactants, or also to competing biosurfactants?
So we are working on Monosule eretritolipids and Cellobiose lipids, and we have several different molecules, as you heard in the presentation. And we're working together with different companies, and the companies had a lot of nice results in different areas. So imagine washing powder, but also for dishwashing and also for cosmetics. So the aim in our project is really that the companies look in their potential. So to find out how can we use this novel surfactants for really a wide range, hopefully, for the market.
Okay, so from the audience, there's a question: is there already a current production of your biosurfactants on Monosule, erythritol, and Cellobiose lipids?
For the Cellobiose lipids, I don't know anybody who is producing it, and for Monosule erythritolipids, I just know one company in Japan. So I guess we are open that we find some partner who would like to produce that, surfactants.
Okay, and also from a market perspective, can these surfactants compete with existing surfactants in price, or do they need to have very specific properties to come into the market?
So I guess everything what is bio-based is more expensive than chemicals what are fossil-based. So in the first step, I guess we need some products what has also a large market or also a higher price. For instance, cosmetic industry is more expensive, I guess, than plastic industry. And I believe that also from the consumer side, they want to have bio-based products and more sustainable products. And I believe also that the consumers are able to, or they are willing also to pay a little bit more if they know that there is inside a sustainable bio-based product.
Okay, one question from the audience that is more or less linked to this. Can these biosurfactants enter the market without any new regulations?
No, so regulation means we have a new chemical, I would say, and here there needs to be registered at the ECHA, so this is the European Chemical Agency, and for the registry they also need some tests according their environmental impact, something like this, and when we are starting, the industry also could produce more than 1 ton, then we also need some higher registration. So so to say, the REACH registration is necessary. So it depends on the production size and the registry. So the first step is really always a registry at the European Chemical Agency.
Okay, you already mentioned that you would imagine to have some kind of a green premium if you put them on the market. The people want to have it bio-based. But on the other hand, do you see any hurdles for social acceptance? Like, for example, in the audience, there's a question: do you see an ethical problem in using agro-based raw materials? Or the other topic can be: do you use GMO-based fungi to produce your materials, and see any problems in this case?
Up to now, we're using wild type strains. That means they're not genetic modified. So in this case, I don't see any hurdles. And on the other hand, if we look on that, what they need to consume, so renewable resources, here it is possible to fetch sugars and also some side products from sugar industry, melasse for instance. And on the other hand, it is also possible to use different plant oils. So they could be from regional areas, so rapeseed oil. We don't need to import soybean oil or also coconut oil or something like this. So I believe that for the production of Monosule eretritolipids, we can also have regional sources and not long transport ways, and so it's more sustainable, I would say, than using imported stuff.
Okay, also from the audience, and directly linked to your last sentence, can you comment on the sustainability of the surfactants obtained by whole cell fermentation? So did you do any LCA on this?
We are collaborating with University Stuttgart in doing the LCA, and our aim in the LCA was first that we optimize the process. So we have a scenario where we do the fermentation, the purification. That means we have different steps from the broth, from the fermentation broth, to the high quality product. And on this way, we have seen that some steps are not that sustainable. And that was the reason why we worked together with the LCA, the university, for doing the LCA, to find out what are the things we need to optimize in the laboratory. And we get so many ideas to enhance the titer, to which kind of raw material we use. And this is a very important thing in this collaboration, to find out how can we really optimize in a laboratory.
Okay, a bit more specific question, but partly you already answered this. Are the biosynthetic enzymes known for these glycolipids? And if yes, have they been modified? Or I would add, is there a chance in modifying them to change structures or improve the yields and maybe also the properties?
I know some literature work at different universities. They are working on modifying the strains to really change the chemical structure. So this is an idea that we produce specific molecules and not too much a wide range. So as you have seen in my presentation, we can have a couple of Monosule eretritolipids with different variations. And this is something what different researchers are working on it. This is that what I know. I don't know any work on optimizing the yield directly with the genetic modification. Here, I guess the most important thing is the process engineering. So to enhance the titer and also to have a robust fermentation, because we have an aerob fermentation. We need to supply oxygen. And that means we have also a lot of foaming problems. And this leads in scaling up to a lot of troubles. And here, the process engineers really need to establish a very robust process.
Okay, so one last question from the audience. If a young startup wants to source these surfactants and optimize a mix for a specific consumer need, for example, household cleaning, whom should they reach out? Do you also mentor these kinds of startups?
Yes, we support the startups, and also a team of my students also considered doing this. So, if young scientists are interested in collaborating and getting some mentoring, they can contact me.
Great. That's a very good answer for the last one. Susanne will also be in the panel discussion. So thank you for now, and we will see you later.
Okay, thank you.